this picture on the left here is one of my favorite pictures. It's the picture of the first transplant that happened in the world. It happened between two identical twins, and it occurred in Boston in the Brigham Hospital where I used to work. And this advancement really helped medicine and pushed a lot of treatments forward. But it wasn't until Dr. Terasaki came along that it really took off. And what Dr. Terasaki did is that he developed the technology to be able to match donor and the recipient's cells, so that in the cell surface, there wouldn't be molecules that would reject the cells of the donor. So this advancement, of course, was fantastic, and it led to the formation of a company that resulted in the endowment and the support from the Terasaki family to build this new institute called the Terasaki Institute for Biomedical Innovation. So our goal here is to really bring in different fields, including material science, to innovate and foster practical solutions that restores or enhances the health of individuals. And I think this is particularly important in this era as we are starting to develop technologies that enables us to dig into the person's genome and proteome and understand how the variability between different people happens. So a quick plug, I definitely encourage everyone to follow us through our social media. We are recruiting a number of people at different levels, so I encourage you guys to go to our website and check for our openings that are happening right now. So what's this all about? What is this engineering approaches and how it can enable this whole aspect of precision or personalized medicine? Well, one of the things that we're trying to do is actually try to build materials and other types of technologies that can do that. So we build a lot of different aspects of materials that can be used for medicine. For example, being able to use glues or other types of surgical materials that can be deformed with the patient itself. We made materials that can be used inside blood vessels to plug blood vessels or make devices inside blood vessels. And we've also used materials to be able to develop new arrays of smart devices. These are materials that can incorporate electronics and hydrogels or other systems to make the existing medical devices more efficacious. So for example, being able to add sensors into wound healing patches, or being able to put different kinds of actuators and sensing technology inside catheters that can navigate blood vessels. Also, we're very much interested in using cells and using personalized cells, whether those would be stem cells or immune cells that can be used for treatment of a number of different diseases. Today's talk, I'm going to focus on a little bit of the sections below. These are how we can merge materials and technologies like 3D printing to be able to address things related to making tissues using technologies like biofabrication, and using these tissues to create systems that we call organs on a chip, or microphysiological systems that are used as bases for developing new drugs and modeling human physiology. So this, of course, all happens in teams. The work that we do is highly interdisciplinary and is on the basis of this convergent science. So everyone comes together from different areas and works in teams to build particular systems that can solve problems. So, and the way material science kind of integrates into this is through being able to build materials that are smarter and can be responsive. So we work a lot in hydrogels as well as other materials, but with respect to hydrogels, we do make them in different shapes and sizes, so we care about how we cross-link them. We can also be able to integrate different types of hydrogels together to be able to get new properties that can allow us to have new features in these materials. So one of the things that we've done is actually started to also combine these materials with different fabrication processes. So we can use a lot of different technologies like molding and lithographic approaches, printing approaches, and microfluidics to control the shape and architecture of these materials. Now, why do we want to do that? Is because we can control the final structure, so we can actually make tissues that are more like our own human tissues. So I'm going to give you one example of how this is used in applications. Well, one of the things that we're interested in is to decrease the cost of making drugs. Right now, to make a single drug, it takes about a decade and a couple billions of dollars to be able to do this. And the reason for that is because the whole drug discovery process is like a funnel. So you start off with lots of drug candidates and you put them through different stages until you find the one that's going to be approved as a drug. Now, this is a very expensive process, and particularly if you fail at the later stages of approval, you've already spent hundreds of millions of dollars in this process. So why does this happen? Well, the reason that happens, that why you fail at a later stage, is because you can't predict you're going to fail at these later stages. And the process is typically you first do stuff in cell culture, and then you do it in rodents and these animal models, and then you go through different population sizes to be able to see whether the drug is safe and if it's going to be effective. Now, the challenge here is that you can't predict whether these animals are going to be effective as good models. And really, this has caused the drug discovery costs to go balloon to this gigantic sizes. Now, when you think about the failure and why it happens, a lot of it is because, as I mentioned, this inability to predict the efficacy and safety of our compounds. And even if when a drug is approved, then you do have a lot of variability between people. So you have cases like Vioxx, where the drug worked, but there was this small population of people that would not be able to deal with the drug, and they would have a lot of side effects, and that caused a number of deaths. So Merck had to recall this drug, and it cost them billions of dollars to do that. So the challenge here is, how do we enable us to predict better drug safety and efficacy models? And the current models of whether it's cell culture or animal models don't replicate the human complexity, the human physiology. So how do we bridge this barrier? So an emerging field has developed in which the idea is to take human cells, build human tissues, and it could be even tailored to an individual patient, and be able to even interconnect these different human tissues so that you get a better systems behavior, kind of like our own body. So these different tissues could be barrier tissues like lung and gut and skin. They could be metabolic tissues like the liver, and they could be other kinds of functional tissues like bone and heart or skeletal muscle, etc. So there's a lot of work that's gone into building these tissues, and material science is an integral part of this. So when you think about some of the current work, there's a lot of advances in trying to overcome the 2D monolayer aspects of this that uses animal cells and our individual cell types to building more complex systems that are more mimetic of our own human tissues. So when you look at these different tissue types, you start seeing some consistencies. They're not just packs of cells, but there's architecture that is associated with their function that we're trying to recreate. And this is why techniques like bioprinting or microsystem engineering is very important for these systems. So let's think about some of these. So for example, in the lung, as you see down here, it's multiple cell types, but also the system expands and stretches similar to our own tissues. So it mimics a lung physiology. So a few years ago, we started to work on these systems, and we started to build systems that are like this. So this is one of our early microphysiological systems. where you see here a lot of displays that are attached to sensors and valving and pumping that controls the fluidics. And inside this box here, this is where you have your tissues and cells located in an incubator. So if you zoom into this box, we have systems like this, where you actually have things that start looking like computer chips instead of moving electrons around, they're actually moving liquids around, fluids that mimic our circulation. So what you have here are these fluidic channels where you can attach different tissue types to it. You can attach different fluidic things like bubble traps and pumps and reservoirs. And you can also attach sensors so you can monitor how these systems behave over time. So what you can see here is a picture of one of the early prototypes. And just to show you how these things work, here's an example of a bubble trapper that you can basically see a bubble enter, and then it gets removed from the feed here so that it doesn't go into where the tissues are. And here's an example of, you can see a sensing module where in real time, we're changing the liquids that are moving into these different channels. So this basically shows how much control we have over these by using these pneumatic pumping and valving systems in our microfluidic channels. So we do develop a lot of 3D printed tissues for these. And what we do is that we make a little reactors, little chambers, and we can open and close these, and we can deposit our cells and tissues using 3D printers into these systems. So here's a couple examples. We can do this either by printing little tissue pieces. This is an example of a liver tissue piece that we can print, or an example of a heart tissue piece. Or we can do other techniques like photo patterning and micro patterning to be able to deposit these cells inside these little chambers. Now, we do a lot of biological characterization of these tissues, and some examples of this you can see here, where we can actually take different liver cell types, hepatocytes and other support cells. We can mix them in the right ratio and make these little micro tissues that are mimicking liver tissue behavior. And there's a lot of biology here, but basically they express the right kind of enzymes and molecules that are expressed by the liver cells. They remain viable, as you can see by the green images here, but also you can maintain their viability over long periods of time. In this case, up to at least a month. Now, what's interesting is that these liver cells are functional, so they predict how our systems behave. So if you add a couple compounds that were initially approved, but then subsequently recalled due to liver toxicity, we can actually get some of that behavior in our system. So here, these are our liver microtissues. As we start adding different cell types, adding different concentration of these drugs, you start seeing a lot of these red cells emerge, which kind of shows the cells are dying. And this is really good because now it allows us to predict some of this toxic behavior. We can do the same thing with other tissues, and I'm going to give one more example of a different tissue, in this case, heart tissue. In this case, what we do is that we actually use induced pluripotent stem cells. These are cells that can be derived from patients, and we can start taking these cells and being able to actually combine them to make little pieces of tissues that mimic heart tissue behavior. And these tissues, again, there's a lot of biological characterization that we do to show that they express the right kind of molecules, they remain viable over time, and they actually beat very consistently and robustly, similar to heart tissue that we have in our own bodies. And what's interesting is the same thing if you add compounds that were recalled due to toxicity of the heart, you can see the same behavior where as we start adding these concentrations of these molecules, you start seeing more of these red cells emerge. And not only that, the beating rates of these heart cells as you start adding these molecules goes down, and the cells do not beat at higher concentrations. So we can use this for these kinds of toxicity analysis. to see what the behavior is. We can make disease models. Just to show you another example, we can also use this for personalized medicine. So being able to take tumor biopsies from patients, in this case, liver tumor biopsy, and put it in these systems, and even have a healthy tissue from the same patient, in this case, being able to take a skin cell and reprogram it into a heart cell, and be able to make these heart tissues, and be able to test chemotherapeutics on these, or different types of drug or immune system drugs, so that we can actually see what the effect is, whether it's going to be working for that individual patient. So there's a lot of data here, but the general idea is that in these systems, we can monitor how the tissues are behaving. We can monitor different things like pH, oxygen, and temperature. We can monitor with little mini microscopes how the cells are behaving. And we can even have little sensors so that when the cells secrete something, we can monitor them. So for example, this liver cells secrete albumin, so we can monitor that, or GST alpha. And based on these, we can actually monitor how this, in this case, the chemotherapeutic is killing the liver cells. We can also do other things like put heart tissue, look at the heart tissue, that's the example of the healthy tissue here, and then see how the cells are, the healthy cells are sensing the chemotherapeutic, and what are the side effects associated with this. So all of this technology is enabled by techniques like bioprinting, so where we can actually build these tissues. So there's a lot of different technologies that we do for these sorts of things, but I'm just going to give you one example of advances that we can make in bioprinting that actually enables us to be able to make these tissues that I've shown you in the previous application. Now, bioprinting has come a long way, so that the dream of printing tissues in the future has come closer to reality, but there's still a number of challenges. So for example, we still have need for better inks. We have the need to make these systems have better throughput and have better resolution. And we need to be able to make the complexity of different tissue types by having multi-material printing capability, so you can put different cells and different materials in different architectures as we build these types of tissue structures. So for today, I'm just going to give one example of the types of multi-material printing that we're doing. And this particular example is a biomimetic example, or bio-inspired example. It actually started off by the need of trying to print different materials and getting inspired by how nature does it. In this case, a spider. What a spider does is that it has a number of different glands in its back that basically it can mix the components of in different ratios, so that it can actually print, or in this case, extrude silk that it wants with different properties. So we can get this kind of inspiration from nature and engineer fluidic systems that mimic it. So in this case, what we have, instead of these different glands, we have reservoirs that can feed these microfluidic channels. And we can control which of these fluidic channels actually gets expressed. So in this case, we can, the different colors in this fluidic channel are actually the different materials, let's say. And the little circles on top are little valves, so that we can control which of these channels is open or closed using the pneumatic actuation method here, where we can have a little chamber on top that's got air in it, and we can pressurize the air so we can pinch off the channels. So we can, by doing this, we can now extrude different materials that are basically coming out of these different channels. Just to show you how this works. Imagine if you have 3 channels where you have 3 different materials, the red material, green material, and the blue material. And if you first open each of these sequentially, then what gets extruded is basically the composition of these in a sequential manner. Now, if all of these are open at the same time, then all of these materials will be expressed in this region of the extruded material. So by doing this, now you have a lot of fine control over what is coming out of this channels over what period of time. So just to show you how this can be advanced even further, imagine if you have the same 3 channels with the same 3 different materials, but now you have regions where you open multiple channels at the same time. So as you start changing these materials, then you don't only change the chemistry along the extruded fiber, but also you can change the topography, because when you open multiple channels, more material comes out, so you have thicker regions, as you see here. There's other engineering mechanisms that you can have. You can actually be able to change the topography, or even have multi-phase systems so that you can actually have air bubbles, oil droplets, cells, all embedded in these extruded materials. Now, it doesn't really take much to start adding these upstream to the printing capability, and this is exactly what we've done. So just to show you an example here, just a simple bi-material printer where you have 2 different channels coming out the same nozzle. And what you can do here is now you can print different materials on demand. So here you can print 2 different materials in 2 different layers. You can even have the 2 different materials expressed in the same fiber of the same layer, and based on that, be able to build different types of complexity that you want. Now, just to show you where we're going with these things, we are now developing these multi-material printers that can have a lot of complexity in what they're printing. So just you can imagine having multiple different inlets where each of these will have different materials, and based on which one of these materials or combinations of them you extrude, now you can start building all kinds of complex structures based on this. And the advantage with these systems is that as you print the materials, there is no period where you actually have to change nozzles. You can print the materials without any delay, and that allows you to speed up the printing and be able to use it more for biological applications, which are very time sensitive. And now we're kind of building different types of capabilities using these printers so that we can actually generate the complexity that's needed as we start to build our tissues. So this is what I wanted to talk about today. Just a quick overview of some of our existing work on 3D printing, but also on application of it in building these organ-on-a-chip models. So with that said, I think that this is a very exciting time to be doing this kind of research with personalized and precision medicine. Normally, what people have thought about is really the big concept on top here, which is uses the omics, the bioinformatics, and the genomics and proteomics approaches to be able to address patient needs. Now, what we're doing is we're starting to really take engineering and material science and applying it to this field by being able to make implants that are tailored to a person, making physiological models that are tailored to a person, making materials that are tailored to the person, and making devices and smart systems that can monitor and respond to the needs of the individual. So I think this is all very exciting field, and hopefully that's what we will enable moving forward at the Institute.